Friends, today we are discussing about the topic disaccharides, its structure and biological role. Before that, myself, Ashwati N, studying first MSc Biology, St. Mary's College, Thrissur. As we know, what is disaccharides are? Disaccharides, which consisting of two monosaccharide units, which is held together by a glycosidic bond, and they are crystalline in nature. They are water soluble, sweet to taste, and one more thing that they are of kind. They are two types: reducing sugars and non-reducing. Based on their presence of free aldehyde or ketone group, they have classified in both reducing sugars and non-reducing. And the examples are maltose, sucrose, lactose, cellobiose, and trehalose. These are the major examples for disaccharide. And first one is maltose. This is the structure of maltose. And maltose is composed of two alpha D glucose units, which is held together by an alpha 1 to 4 glycosidic bond, and there is a free aldehyde group which is present on the first carbon of the second glucose, which answers the reducing reactions besides the oxazone formation. And maltose can be hydrolyzed by dilute acid or enzyme maltase to liberate two molecules of alpha D glucose. And isomaltose is a structure, is another term which is the glucose units are held together by alpha 1 to 6 glycosidic linkages. And its biological role of maltose, which is used in brewing industry, also in duff with strong fermenting yeast, as they are able to metabolize quickly, and it is used in infants food. Second one is sucrose. And sucrose, we know it is a cane sugar, and mostly which is produced by sugar cane or sugar beets, and it is extremely abundant in plant world, and it is familiar as table sugar. It is made up of alpha D glucose and beta D fructose, and these two monosaccharides are held together by a glycosidic bond between the first C1 of the alpha glucose and the C2, the second carbon of beta fructose. And unlike most disaccharides and oligosaccharides, sucrose contain no free anomeric carbon atom. Because these two anomeric carbons of the two hexose are linked to each other. For that reason, sucrose does not undergo mutarotation, and it does not react with the phenyl hydrazine to form oxazone, and does not act as a reducing sugar. It is an important source of dietary carbohydrate. It is sweeter than other common sugar, namely glucose, lactose, and maltose. And it is a sweetening agent in food industry. And sucrase enzyme, it is an industrial enzyme which hydrolyzes sucrose to glucose and fructose, which are absorbed. And inversion of sugar is another process with which sucrose is such dextrorotatory, and when it is hydrolyzed, sucrose become levo. Process of changing its optical rotation from dextrorotatory to levorotatory is referred as inversion, and the hydrolyzed mixture of sucrose containing glucose and fructose is known as invert sugar. And the process, which is the hydrolysis of sucrose by the enzyme sucrase, and it liberates one molecule of each glucose and fructose, and sucrose first split into alpha D glucopyranose and beta D fructopyranose. But beta D sucrose first split into alpha D glucopyranose and beta D fructofuranose, and beta D fructofuranose is less stable, and immediately it gets converted into beta D fructopyranose, which is strongly levorotatory. So that overall effect is that dextrosucrose on inversion is converted into levo form. And its biological role is important component of soft drinks and other beverages, and it serves as an antioxidant, widely used as a food preservative, and used in many pharmaceutical products. Lactose, next one. And it is a milk sugar, commonly known as milk sugar. It is a disaccharide which is found in milk, and composed of beta D galactose and beta D glucose, which held together by beta 1 to 4 glycosidic bond. And the anomeric carbon of first carbon of the glucose is free, hence lactose exhibit reducing properties and form oxazones. And lactose milk is the important carbohydrate in the nutrition of young mammals. It is hydrolyzed by the enzyme lactase to glucose and galactose. Its biological roles are used in food and pharmaceutical industry, main carrier for drugs, because it's not sweet, safe, and available in highly refined form. It makes good quality of the tablets. In food industry, it is used based on its relative sweetness and being a source of energy, less sweeter than sucrose. And next one is cellobiose. It is a disaccharide. It is a reducing sugar, and a repeating disaccharide units of cellulose has beta 1 to 4 glycosidic linkages. It is an identical structure with maltose, except that there are linkage is beta 1 to 4 glycosidic linkage. And cellobiose is formed during the hydrolysis of cellulose, and they have a free anomeric carbon, so that they have reducing properties. And biological roles are, they can humans can derive cellulose from cellulose laden foods such as honey, corn syrup, certain fruits and vegetables. And the presence of cellulase helps in the breakdown of cellulose into shorter chains of glucose units such as cellulobiosis. These are the biological roles. And the next one is trehalose, which contains 2D-glucose residues, an example of a non-reducing disaccharide in which two anomeric carbon atoms are joined. It is the major sugar found in the hemolymph of many insects. And the biological role, it stabilizes the cell membrane, and it particularly useful for the preservation of sperm, stem cells and blood cell, which occur in organisms capable of surviving extreme dehydration, and is used for freeze-drying method of cryopreservation. So that's all about. Thank you.